Hello, hello, and welcome back. So today we are going to be doing a lab on pH. So today's aim is how can we use indicators to determine pH levels? So I do want you guys to write down the definition of pH. So what exactly is pH? Well, remember this is a measurement of the concentration, which just means the amount of hydrogen ions. And whenever we're talking about hydrogen ions, we're going to represent that as H+. All right, so how can we use indicators to determine the concentration of hydrogen ions in solutions? Let's find out. Before we get started, I want you guys to answer these 3 do now questions. Number 1 is, what is the purpose of using indicators in biology? Number 2, fill in the chart below by naming the indicator used for the following biomolecule and describe what a positive test looks like. So if it's a positive result, that means the nutrient being tested for is present. All right, and the third question is, define each group on the pH scale. So pause this video, answer those 3 questions, and when you're ready to go over the answers, press play. So question number 1, what is the purpose of using an indicator in biology? Well, I hope you remember from our lesson on indicators that this is to determine what nutrients or biomolecules are present in a solution. To determine what nutrients or biomolecules are present in a solution. All right, number 2 was asking about 4 different indicators that we talked about last week. So the first one indicates the presence of glucose, and the name of that indicator is going to be Benedict solution. All right, Benedict solution starts out blue, and if it's a positive test result, you're actually going to get a gradient of colors. And that's because remember that Benedict solution requires heat to activate it. Okay, so you might get a test result that is red. You might get a test result that is orange. You might get a test result that is yellow. And you might even get a test result that is green. All of these are still positive test results. They still show that that nutrient is present. But the different colors, or the different colors represent a gradient of how much glucose is present. So if it's red, that means that there's a lot of glucose. If it's green, that means that there is not a lot of glucose. So it changes colors on a gradient depending on how much or how little glucose is present. All right, the second indicator that you should have written down indicates the presence of protein, and that is going to be something called Biuret solution. And remember, Biuret solution is the one that we made by combining KOH, potassium hydroxide, and CuSO4, which is copper sulfate. So again, Biuret solution starts out blue, but if it's a positive test, if protein is present, it's going to turn a violet color, like a purple. All right, the third indicator we used was something that detected the presence of starch. All right, you might remember that that is called Lugol's solution, or just iodine. So iodine starts out amber. I'm gonna use a different color for that. Starts out amber, which is like this reddish brown color. And if starch is present, that indicator is going to turn black. And the final indicator that we talked about last week is used to detect the presence of lipids. And that's something called a brown paper test. Okay, so if it, initially, brown paper is what we describe as opaque. Remember, it was just that brown shade, and it means it's opaque, meaning that no light can get through. It's just a plain old brown paper bag. But if lipids are present, we see that grease leave a stain on the brown paper, and we call that result translucent, which means that light can pass through that bag. Now, it doesn't mean transparent because it's not see-through, it's not clear, but it is translucent, meaning that light can pass through. So the lipids do leave a stain on that bag. Okay. And finally, your third question was to define each group on the pH scale. So whenever you see this scale, this is indicating the level of a pH. So it shows you a gradient of the concentration of hydrogen ions. Okay, so if we have something, if we have a pH that ranges from 0 to 6, that's going to be what we call acidic. And that means that there is a high concentration of hydrogen ions. Okay, if we have a pH of 7, right here directly in the middle, I'm going to do that as purple. That's going to be neutral. And if we have a pH that ranges from 8 to 14, that's going to be basic or alkaline. And that means that we have a low concentration of hydrogen ions. All right, so today we're going to be working with this pH scale and identifying the different pH values of different solutions. So let's get started. First, we've got to read a few passages on what we're actually going to be doing today. So the first part is methods for testing pH of liquids. Every substance around you, from the orange juice you drink and the water you use to bathe, to the saliva in your mouth, has a pH level. Testing the pH of liquid, which can be done in different ways, tells you whether it is acidic, alkaline, or neutral. On the pH scale, which runs from 0 to 14, 7 is neutral, anything below 7 is acidic, and anything above 7 is alkaline. Remember that alkaline also just means basic. So your first question that I want you to pause this video and answer is, why is it important to test for pH? Why do we need to know this? So pause this video and then press play when you're ready to go over it. So why is it important to test for pH? Well, it's important to know how acidic or how basic solutions are. Okay, so we've talked about how enzymes function best at a specific pH. So anything above or below that value, that pH isn't going to work as well. And that same methodology applies to really everything. So some examples are, we want to make sure that the water we're drinking is actually neutral and is not an acid, right? That's not going to be good if you mix up water and hydrochloric acid, right? Another example is we often test the pH of soil to make sure that plants are growing in the best environment possible. Okay, so some examples, we want to make sure our water is neutral. And we want to make sure that soil, let's say, test soil pH to ensure maximum plant growth. Okay, so those are just two examples of why is it important to test for pH, right? We want to make sure that everything is working as it's supposed to and things are running as planned, right? We want to maintain that homeostasis. All right, so I'm actually going to give you guys an example of a naturally occurring indicator. So this is not a chemical that's been man-made. This is something that we actually find in nature, and that is red cabbage juice. So one interesting method of testing the pH of a liquid uses red cabbage juice as a pH indicator. When the cabbage juice is mixed with different liquids, the solution changes color thanks to a pigment in red cabbage called flavin, an anthocyanin. So I'm going to highlight that. So the solution that's not a highlighter. The solution changes color thanks to a pigment called flavin. I also want you guys to note that the reason this pigment changes colors is because a pigment is a protein molecule. Okay, so pigments are proteins. If the color changes to pink, the pH is 1 to 2. If the color becomes dark red, the pH is 3 to 4. If the color changes to violet, the pH is 5 to 7. If the color changes to blue, the pH is 8. And if the color changes to blue-green, the pH is 9 to 10. If the color changes green to yellow, the pH is 11 to 12. So that's a lot of different numbers and colors, right? But the main thing I want you guys to know is that if the color or let me read this last sentence. Highlighter. All right. So this means that pink and dark red solutions are acidic. Violet solutions are either acidic or neutral. And blue, let's use that color. Blue, blue-green, and green-yellow solutions are alkaline. So we're actually going to do this right now. I've got some lovely smelling cabbage juice. So as you can see, the initial starting color is purple. So I actually want you guys to write that down on your paper. Okay, so cabbage juice, oh, that's highlighter. One of these days I'll get these things down. All right, cabbage juice is naturally purple. So I just got this fresh out of the blender. Got some lovely purple cabbage juice. And today we're going to be adding both an acid and a base to the solution to see what happens. The acid that we're going to add is hydrochloric acid, HCl, and this is an acid. Okay, the base that we're going to be adding is actually one that you've seen before, and that is KOH, potassium hydroxide. So this is a base. So I'm going to unshare my screen so you guys can see what's happening. But I do want you guys to record these results as I walk you through this. Okay, so as you can see, we're starting out with some purple cabbage juice. But the first thing I'm going to do is I'm actually going to add some acid, hydrochloric acid, so HCl. And you can actually see that it has corrosive written on it, right? So this is actually a super dangerous acid. It will eat through your skin if it touches you. So let's hope that I don't drop it. Okay, but I want you guys to see what will happen once I add this acid to this cabbage juice. Okay, so I'm gonna start mixing it around and you start seeing that color change. And add a little bit more. All right. So you can see, hopefully, that as I added the acid to this, there is still some at the bottom just because I've got some of my solid particles have settled there. But you see that that red cabbage juice actually turned a reddish pink color. So I do want you guys to note that when I added an acid, the cabbage juice turned red. All right. But now let's see what happens once I add KOH, which is my base. Okay. You're actually about to see two things happen, so make note of both. So first, when I add some KOH, oh, I added too much. All right, so you kind of see this happening. You actually see, if you ignore that green part, that this, no, that the solution actually turned purple again. And I think I added a little bit too much too quickly, so you might have missed that purple. But the reason it turned purple is because we actually just neutralized that acid. So you can see that it's kind of purple again. That's because we added the same amount of acid and base. So it actually neutralized and went back to a pH of 7. But if I keep adding this base and add a little bit more, you can see that it actually turns green. So I'm going to stir it a little bit more. In fact, I'm going to stir it around using a straw. There it is. So you can see that now we're actually dealing with a green solution, which means that we just added that base. So this solution is now basic, and our naturally occurring indicator showed us that that's correct. All right, so go ahead and write both of those results on your paper. I'm going to flip back to my paper and write the same as well. Okay, so you should have already written that cabbage juice is naturally purple. And I want you to write what happened when we added the acid and when we added the base. So when we added the acid, the solution turned red. But when we added that base, the solution turned green. Okay. I want you to also note that when we have the same amount of acid and base, we actually ended up neutralizing that solution, which means it returned to a pH of 7. All right. So that's all you need to know about red cabbage juice as a naturally occurring indicator. But now we're going to be talking about some not so naturally occurring indicators. These are in fact man-made indicators used to test pH. We're going to be talking about 2 different types of pH testing strips. So read along through this passage with me. pH testing strips contain a series of indicator bars that each change color after exposure to a solution. I'm going to decrease that. All right, so they change color after exposure to a solution. The strengths of the acids and bases on each bar differs. So what we're going to do is we're going to collect a sample of liquid in a clean container, ensuring the sample is deep enough to cover the testing strip. We're going to dip a strip into the sample for a few seconds and wait for the indicator bars on the paper to change colors. We will then compare the end of the test strip with the color chart that came with the paper to establish the pH level of the liquid. So we're actually going to be working with 2 different types of pH testing paper today. The first one is called litmus paper. Now with litmus paper, we're actually dealing with both red and blue paper. So we have to use both of them together. So when these red and blue paper strips are used, they will determine the pH levels of a solution. But I do want you guys to note that litmus paper only tells us if the solution is an acid or a base. So it only tells us if the solution is acidic or basic. But we have to use both of these strips together. So I'll show you what those results are going to look like. So when we start out with red paper and we start out with blue paper, okay, if the red paper remains red and the blue paper turns red, so both of those papers are red, that tells us that that solution is acidic, which means it has a pH ranging from 0 to 6. Okay, and we're going to be representing acids as H+. Oof, I can't write. Oh, I thought that whole thing disappeared. I was about to vomit. All right, acidic, okay? But if we're using both of those samples, so again, we have red paper and we have blue paper, but instead of turning red, we actually see that red paper turn blue and the blue paper remain blue. That tells us that the solution is basic, which means that it has a pH ranging from 8 to 14. And we're going to see our bases represented as OH negative. Okay, so that's why when we saw up here, HCl, that's an acid, and KOH, that's a base. So anytime something ends in OH, that means it's a base. And usually when things start with an H, when we're talking about molecular formulas, that's going to be an acid. Okay, so litmus paper, again, you have to use both red and blue together to get that solution. If we see both of these turn red, that means it's an acid. If we see both of these turn blue, that means it is going to be basic. But we also have a separate second type of pH testing strips called p-hydrium paper. And p-hydrium paper are, again, paper testing strips that indicate a pH level in a range of a color according to a pH scale. So on every container of p-hydrium paper, it actually comes with a scale on the side. And this is because p-hydrium paper is actually special in the fact that it can tell us the exact numerical pH value. So while litmus paper just tells us if something's an acid or a base, P. hydrium paper can tell us, okay, this solution has a pH of 4, or this solution has a pH of 7. So it tells us the exact number of that pH. So when you're looking at this, you'll always use the scale on the side of the container, but anything that turns from a red to an orange so red to orange is going to be an acid. Oh, wow, things just really do not want to work out right now. Please still be there. There we go. Okay, we only lost a little bit. So anything that turns from red to orange, I'm leaving that, is going to be an acid. Anything that turns yellow is going to be neutral. And anything that turns from a green to a blue is going to be a base. Again, but you are always going to use this scale to determine the exact numerical value of that pH. So let's do it. Today we're going to be working with 3 different solutions. We're going to be testing soapy water, but I actually want you guys to correct that because we're going to be using detergent instead. We're going to be using distilled water, and we're going to be using my favorite, black coffee. I'm just gonna get one last sip in. Okay, so what we're going to do, I'm going to stop my share. Okay, we're going to run each of these tests for each of our solutions. So let's do it. What I'm going to do is I'm going to lay out this sheet of paper, and this is where I'm going to put all of my results on. But before I start grabbing this paper, it's super important that I do it correctly. So whenever we're using litmus paper, we're always going to use forceps to grab a sheet of paper. Because if I just grab a piece of this paper with my fingers, that means that anything my fingers have touched since the last time I washed my hands, namely just oils produced by the skin, are going to end up on that paper. And that's going to essentially be cross-contamination, and it's going to impact our results. So we always use forceps to ensure that we're getting an accurate read. Okay, so I'm gonna grab a piece of this red litmus paper. I'm gonna stick it down on my paper. And I'm gonna go ahead and lay out all of my papers just to make this a speedy process. Hey there. You're good. I'll be done in like 10 minutes if that's okay. All right, and I'm also going to lay out a piece of this P. hydrium paper. All right, and the first solution we're going to test is our soapy water, or our detergent that we're going to use today instead. So I've got a vial or a test tube of my detergent, and I've got a straw that I'm going to just use to drop some bits of this solution onto each of my papers. As I do this, I want you guys to record the results on your document. Okay, so what happens when I put detergent on my red litmus, on my blue litmus, and on my P. hydrium? I'm actually going to put a little bit more there. All right. So I'm going to let that sit for a second, and then we will record those results. So you might already be seeing what's happening. So right here, this was my red litmus, which you can see is no longer red. This is my blue litmus in the middle, and this is my P. hydrium on the right. So I want you to go ahead and record that. As you can see, my red litmus turned blue, so please record that on your notes. My blue litmus remained blue. Figure out what that means. Using those 2 papers together, do you think this is an acid or a base? Record those results as well. And finally, record the color of that P. hydrium paper. All right, now after you've recorded those colors and whether or not you think it's an acid or a base, we're going to use our scale to compare this and figure out the actual numerical value of that pH. So if we use this piece of pea hydrant paper right here, and we use our scale, we can actually figure out, oh no, that the pH of this detergent is a 9. So if we use those colors, we can see that that pH is a 9. Right? So please record that as well, that the pH of that detergent was a 9. Now, is that an acid or a base? Well, since it is above 7, that's going to be basic. So you should have recorded that as well using your litmus paper. Because we had both strips of paper turn blue, oh, fallen soldier, that is a basic solution. All right, so that's all you need to know for soapy water. Next up, we're going to do the same test for our water. So again, I'm going to lay out my strips of paper. I'm going to get my red litmus. I'm going to get my blue litmus. Green. Okay. And I'm going to get a straw, and I'm going to just dip a little bit of this water, H2O, onto each of these strips. So I've got my red litmus, my blue litmus, and my pea hydrant. Let's see what happens when I drop my water. All right. So I want you guys to go ahead and record the color of each of these. So right here, we've got my red litmus. Here we've got my blue, and right here we've got our pea hydrant. So go ahead and record those color results. So our red litmus remained red, our blue litmus changed blue, and our pea hydrant stayed a yellow color. Now using that information, I want us to compare those results to our scale. Right, using that information, using this scale, I can see that my pea hydrant paper gave us a read of a pH of 7. So that water has a pH of 7. Please record that as well. Okay, that is all you need to know for water. Now, moving on to the last test, we've got, of course, my favorite, black coffee. Now, usually we shouldn't drink our materials in lab, right? But it's just coffee, and I just got this out of the pot, so we're good. So once again, we're going to lay out my 3 pieces of paper. We're gonna get our red litmus. We're gonna get our blue litmus. And we're gonna get our pea hydrant. Okay. So last but not least, let's put a little bit of coffee on these test results and see what happens. Okay, so we're gonna put coffee on the red paper. We're gonna put coffee on the blue paper. Oh, it's trying to go with us. And we're gonna put coffee on the pea hydrant. All right, I'm gonna give that a few seconds to change colors, and we'll record those results. All right, so our pea hydrant paper is still changing, but we can go ahead and record the results of our litmus paper. So both our red and our blue papers turned red. So if both of them turned red, what does that tell us about coffee? Is it an acid or a base? All right, now, Our P. Hydrian paper turned this nice orange color. So when I compare that to our P. Hydrian scale, that tells us that this coffee has a pH of 5, as you can see right here. pH of 5. So go ahead and record those results. And I'm going to record them on mine as well, just to make sure that we are all on the same page. All right, so to recap, our soapy water, let me pull it out, was this one right here, okay? So both our red and our litmus paper turned blue, which tells us that this is a base. Our P. Hydrian paper turned a greenish blue, and that told us that it had a pH around 9. Our distilled water, our red litmus stayed red, our blue litmus stayed blue, and that told us that it is neutral. Our P. Hydrian paper remained yellow. Let me see if I can pull it up. Right here. Which told us that it had a pH of 7. And finally, our last test was black coffee. Both our red and our blue litmus, I made a mess, turned red, which told us that coffee is an acid. Our P. Hydrian paper turned orange, which told us it had a pH of 5. So again, soapy water is a base, distilled water is neutral, and black coffee is an acid. All right, so that is your pH lab. We are officially done. So what I want you guys to do is now open up your exit ticket. This is your lab report. This is where your grade is going to come from. So please put as much effort as you possibly can into this pH lab exit ticket. This is where your grade comes from. Answer these questions, press submit, and you are good to go. Have a wonderful afternoon, and I will see you guys later.